Hello dear friends, and welcome to this video today in our YouTube channel. In this video friends, we shall be looking at the secret weapons you should use to score 100 over 100 in the biology practicals. Guys, you heard me right. I said 100 over 100 guys, because it's super easy to score those marks, as well as it is also very easy to lose all the marks guys. Even your teachers guys will not tell you all the secrets I'm telling you, because none of them also told me when I was taking my own biology exams. So guys, stay tuned, watch the video from start to finish, because at the end of this video guys, you shall discover quite a lot of things that that you were doing and you ought not to do them, because they are going to ruin all your chances of scoring all those marks. Guys, keep in mind that if you want to have an A grade in biology, you have to work on paper 3 very hard guys. It took me months to actually compile all these points for you guys and to explain them very well guys. So please consider subscribing to this channel, and most importantly guys, please give this video a thumbs up. Just please press the like button beneath this video, because that goes a long way to motivate me. It tells me that you found this content useful, that my months of gathering all these points were not in vain. Okay, thank you so much guys, and put it in the comment section if you enjoyed the video or not. Thank you very much, and if you have any question, leave it as well in the comment section, and I'm going to see you as soon as possible. Thank you very much guys, and share the video with your friends so that they can have access to good content like this to prep for their own exam. So guys, let's dive straight into the reason for this our video today. But friends, before we proceed, I want us to take about 10 seconds to look at the structure of how biology paper 3 looks like. So friends, for biology paper 3, we should know that it has 4 questions. Each of those questions are 25 marks guys. Each of them, you have it for them, but 25 marks, you have 4 questions. So guys, the first question is based on classification. That is all what is being treated in lower seed. Second question as well, analysis of the specimen. Still, it is what is meant to be treated in lower seed. Then guys, the third question we have here is what is dissection. Good. Of which you can be asked to dissect either a bird, a mammal, a fish, or a frog. Though the frog and the toad are not so frequent in the Cameroon GC. Then we have the question 4 guys, which is on experiments. Good. So guys, these are the 4 questions that you have. You are going to have arranged the questions in order, because the first question must be on classification, the second must be on analysis of a specimen, then question 3 must be on dissection, and the question 4 must be on experiment. So as you're entering the hall, you expect to see the exam structured in this manner. So guys, let's dive straight into the major and the most important reason for this video today. Good. So friends, let's now see how you can score 100% in the biology paper 3 guys. Let's start with the first that's the first 2 questions guys, which is based on the paper 1, which is based on question 1 and question 2 guys. Let's look at question 1 and question 2, which is on classification and analysis of specimen. So guys, you should never forget guys, never forget, this is very, very important friends, very, very important. Never forget to start the names of the phylums, class, and the common names with capital letters of all the specimens guys. All the specimens that will be provided for you, sometimes they can give 8, sometimes they can give 10. Make sure that when you're being asked to write the names of the phylums or the major group, the name of the class or the subgroup, and the common name, sometimes scientific names as well guys, don't ever forget to start each with the capital letter, and be mindful of the spellings guys. Take note of the spellings, don't ever spell them wrongly. Practice the spellings well guys. Practice them as if you're reading but a book. Practice and be serious about them, because the Cameroon GC is arranged in such a way that if you fail any spelling, or you spell it correctly, or you don't start with the capital letter guys, you fail across. And it is arranged in a column such that if you fail one name or one phylum, one class, you fail everything on that column guys. That's what I'm saying, that it is very easy to score everything as well, also very easy to lose everything. Good. And guys, also note this particular hint guys, note it very well guys. You should most of the questions, they are going to tell you to either write the common name or the scientific name guys. They will ask to write either the scientific names or the common names, but guys, they can ask you for both these names guys. But I want to advise, and take it from me guys, take it from me, don't ever go for the scientific names guys. Don't ever go for them, okay? Don't ever, rather go for the common names, okay? take the common names. The reason for doing so is that it is very delicate to write the scientific names, guys, and it can lead to inaccuracy sometimes. It consumes your time, and it also makes you not to be accurate. There are specific rules that you must implement when writing the scientific name, guys. Specific rules. One of such rules include that when you're writing the genus name, you must start with capital letters, and once you're done writing, and the species name must begin with a small letter, and you must underline them separately. If you forget any of these rules, guys, you fail everything across, and it is very difficult to master scientific names than mastering common names, right? Because the common names are the frequently called names that we all know. Great. So friends, avoid the scientific names, guys. Avoid them. Rather go for the common names. You're going to score the same mark as someone who went for the scientific names. Good. So friends, don't forget to like this video if you found it useful, and consider subscribing to our channel if you want to have all the latest updates. So guys, we've talked about how to write the names of the specimens. You can also be asked to give diagnostic features in place of the common names, guys. Of course, you should not write that, right? Good. So friends, let's look at the habitats of the specimens. Good. We have the habitats, and guys, you should never use words like aquatic and terrestrial. This is the most mistake that most students make, and it kills their marks, guys. Oh my god, I feel like crying, guys. These are the mistakes that candidates make, and they keep scoring zeros on the same section, guys. Never use words like aquatic, never use words like terrestrial. Let's take for instance, guys, if you're being asked to give a habitat of a fish, right? Don't say aquatic. Of course, we all know a fish is an aquatic animal, right? Good. But you should rather say it lives on what? In seas, oceans, lakes, and so on and so forth, guys. Don't ever mention aquatic, even though aquatic is correct, but it is not accepted in the Cameroon GC board. Good. And guys, don't mention terrestrial. If you're being asked, ask the habitat of a grasshopper, of course we all know it's a terrestrial organism, right? Or better say the habitat of a guinea pig. Of course we all know all of them are terrestrial organisms, but for the grasshopper, guys, I expect you to rather say it lives in bushes or on farmlands, okay? Then for guinea pig, it lives in what, guys? It lives in dark kitchens, right? Good. So you should be specific. Once the examiner sees aquatic, sees terrestrial, they mark it wrong without thinking. Okay, they mark it wrong without thinking. So never use aquatic, never use terrestrial. Rather be so specific about the exact location of those organisms. Great. Then guys, for the ecological relationships, guys, you should always try to link all the specimens concerned if possible. You can be given about 5 specimens, and you're being asked to construct ecological relationships about those specimens, guys. For instance, they can give you a toad, they can give you hibiscus flower, they can give you mushroom, and you're being asked to bring forth ecological relationship that exists between these organisms, guys. You can start, for instance, by saying that that the mushroom and the toad release what? Carbon dioxide, which is used by the hibiscus for what, guys? For photosynthesis, right? Great. So you should try to link all the organisms together, their functions and their role in the habitats. And you can also in turn say that the hibiscus flower releases what? Oxygen, which is being used by what? By the mushroom and the toad for what? Aerobic respiration. You can also start by saying that when the hibiscus flower, when the toad die, the mushroom help to do what? Help to decompose their dead bodies, thereby clearing the ecosystem, right? And so on and so forth, guys. You can also say that the mushroom and the hibiscus can serve as food for what? For the toad. So guys, this is how you can bring forth ecological relationship and score the marks, okay? Good. So guys, alright guys, you should never forget that you should so guys, you should never forget that the mushroom is a decomposer, okay? Never forget that the mushroom is a decomposer. If you've been asked to construct that relationship between organisms and you find the mushroom there, guys, don't ever forget to include this very important rule, because the main reason why the examiners will be including the organism mushroom is if you know that it's a decomposer. Okay, if you know it's a decomposer. So friends, don't ever forget that. You should also know that it releases oxygen into the atmosphere, which is being used by plants for photosynthesis. And you should also say that if there are insects there, guys, if there are insects there, most insects provided, guys, will always most insects provided, guys, have effects on plants for pollination. Like for instance, if a honeybee is present, guys, you should rather say that if a honeybee is present and a plant, or that's a flowering plant specifically like hibiscus or like morning glory, you should tell them that the honeybee helps do what? Helps to pollinate the flowers of the hibiscus, thereby assisting in what, guys? In reproduction. So friends, in other words, what I'm trying to say is that if you're being asked to bring forth ecological relationship, guys, you should rather try to look at it. You should try to observe the reproductive role, the respiratory role, the feeding relationship, and what, guys, and you can talk in terms of what is shelter. Okay, because the hibiscus flower sometimes, guys, can serve as what? As a habitat for what? For the honeybee sometimes. Okay, it can serve as a habitat for it. Good. So guys, let's now talk about the dichotomous key. Good. Let's now talk about the dichotomous key, because it's super important as far as the biology practicals are concerned. So guys, you should always use features of organisms that can be seen, guys. It was supposed to be an N here, friends. Always use the feature of organisms that can be seen. You know that a dichotomous key is a branching key. Okay, it is used to separate specimens using their observable visible characteristics. You should never use a characteristics that is not, that you cannot see. Okay, like for instance, they might ask you to distinguish between a toad, a mushroom, and a hibiscus, right? You can start by bringing forth the one that is very easy, right? You can start with a mushroom, and you write under the mushroom that presence of what? Presence of a pilus. Okay, presence of a pilus. Then you come under hibiscus and toad and say that what? Absence of a pilus. Okay, good. You take the next now. You can take for instance a toad, right? You write under presence of what? A rough skin, and you come under hibiscus and say what? Absence of a rough skin. Just like that, guys. Just like that. So guys, I hope it was useful, right? So friends, for a mushroom, you should rather use the very easy observable features such as the stipe, the pilus, the basidia, which no other organisms have. Okay, which no other organisms have, guys, have. Good. So these are the three structures that are so visible for the mushroom that you can always use to distinguish it from other organisms. Good. So friends, and take it very important, guys, note, this is super important, friends, note that color and size are not considered when classifying organisms, because, guys, I'm sure you must have heard this from your teacher, right? You must have heard it from your teacher. If you haven't heard it, guys, know us from today. Don't go to the hall without knowing this plot point, guys. It is so done because the color and the size are simply features that vary continuously with time. Okay, they vary continuously with time based on the feeding habits of the organisms and so on and so forth, guys. You can take for instance guys, a chameleon, right? The color of a chameleon, as you all know, guys, changes with respect to what? Its environment. So you cannot say that a chameleon is blue in color, is reddish or whatever. You cannot say so. Okay, so the color of the chameleon depends on the color of its environment. So it tries to camouflage, to blend with that of its environment, and sometimes it can also camouflage to escape from predators. Good. And a snake as well, guys. We also know that the size of a snake sometimes is very difficult to determine, because sometimes the snake might swallow a very large organisms and it will increase in size, of which after it must have consumed all those organisms, all the organism, guys, its normal shape comes. So you cannot actually predict its size. Great. So friends, let's see how we can compare now two organisms, like to distinguish or to give their similarities, guys. Let's look at them. Okay, friends, let's see if we can compare two organisms. Guys, you should know that, always know that the word compare, guys, the word compare simply means you should give both similarities and their differences. Good. The word compare, guys, is super important because each time the exam says compare, what I've actually seen most students do is that they always just give only the differences and end there. Okay, so don't do that, friends. Don't do that. Each time you hear about the word compare, talk about both similarities and differences. So guys, let's look at the frequently asked organisms that that the GC board always brings for that we should compare them, guys. Let's look at the frequently asked organisms. So guys, the organisms include, we have the dogfish and the tilapia, we have the woodlouse and the crab, we have Selaginella and Lycopodium, we have the moss plant and the liverwort, we have the toad and the frog, we have the hind and forelimbs of the toad or the frog, we have the tadpole and the frog or the toad. So friends, before you enter into the exam hall, make sure you know how to distinguish between these two organisms. Always make sure you know how to distinguish between them. Okay, always make sure to distinguish them, especially guys, especially Selaginella and Lycopodium, because sometimes their differences are very difficult. Okay, it's very difficult to come up with their differences and their similarities. Okay, so always know how to differentiate between them and also bring forth similarities. Always know, guys, always know, it's super important. Okay friends, let's not talk about the trophic level now. Let's talk about the trophic level of organisms, because the GC board always brings that, always brings it. And guys, it's super important. You should never forget this, guys. Never forget this. Never forget. Okay, I've put 2 stars because it's super important, guys. It's very, very important. So friends, you should know that all producers are what green plants that can photosynthesize. Okay, any green plant that can photosynthesize, guys, is a producer. Because when it's a trophic level, they either want you to classify it as either a producer, a primary consumer, or a secondary consumer. Sometimes you can talk about the tertiary, quaternary, and so on and so forth, guys. So but we'll talk about only the producers, primary consumers, secondary consumers, and the decomposer, because this is what the GC board will always be asking you for. So guys, you should also know that primary consumers include what? Herbivores and what? Animals that can feed on producers. Good. So guys, all the herbivores, animals that feed on these producers, or animals that feed on herbs or trees, or animals that can feed on these producers we just listed here, are what? Primary consumers. Good. So friends, you should also know that secondary consumers are mostly flesh eaters, that is animals that can feed on primary on producers as well, guys, as well. Okay, that's animals that can feed on producers as well. So friends, if you can see so well, right, you will see that human beings are what? Are both. You can classify a human being as both secondary consumer and a primary consumer, right? Because we both feed on producers, and we also feed on what? On animals that feed on these producers. Okay, we also feed on producers, and we also feed on herbs, right? Good. So friends, also know that the decomposers include what? You have animals, or you have organisms that feed on the remains of what? Of these decayed producers, secondary consumers, and primary consumers. Okay, things like mushroom, things like maggots. Good. Maggots are the larval stages of what? Of the fly, right? There is the larval form of a butterfly. Good. Sorry friends, is a larval form of what? Of the housefly. Good. I'm sorry friends, it's a larval form of the housefly. So that's the maggot. Good. So friends, let's now, friends, let's look at the question 3, which has to do with the dissection, guys. As you can see, friends, I've written the question 3 and I've put how many stars, guys? 1, 2, 3, 4, 5, 6 stars, guys, and this red star 7 in the middle. The reason for this red star, guys, is that this dissection is a very easy area to pass, but most students always fail. This is the meaning for this, just 1 red star. Then now you have these 6 yellow stars, is to tell you that there is a huge remedy for this same question, which I'm going to be unveiling now. So friends, if you've not subscribed to the channel, guys, please go back and subscribe to the channel, because we shall be uploading so many important contents that are going to help you so well in your exams, guys. Don't forget to watch the video from start to finish before you go for any exams, guys. I'm so sorry that this video has been released when some students have done their biology practicals already, guys. I highly apologize for that. Just that I'm also busy. Okay, I'm also busy sometimes. So friends, you should note this. Note is super important, friends. You should note, guys, you should never spend up to 30 minutes dissecting. Okay, never spend up to 30 minutes dissecting. Why, friends, is that the dissection itself carries just a maximum of about 7 marks. Okay, 7 marks. Keep in mind that each question in this paper is what, guys, is 25 marks. Okay, each question you answer is 25 marks, so the dissection carries just about 7 marks, and the rest of the 18 marks, guys, is for the well labeled and annotated diagrams, because most students always feel so excited about this dissection and they tend to waste a lot of time dissecting. So friends, never spend up to that time. Okay, never spend it. It should be a maximum of 30 minutes. 30 minutes is even too much. Okay, very too much, because it takes just about 7 marks and the rest of the 18 marks are left for the diagram. So I suggest you mostly spend the time trying to draw good diagrams, guys, and annotating them properly. And friends, you should, like I said, guys, you rather spend majority of your time drawing good labeled and annotated diagrams. Good. And friends, don't forget that to annotate a feature, guys, like for instance, you can be asked to draw a fish and or better still, you can be asked to dissect a bird and display the digestive system. So friends, and you can be asked to annotate two parts that are being used for chemical digestion, right? For chemical digestion. So we can come under, we can or better still, yeah, great, or we could still label the liver, right? We could label the liver and say what, and say it is J shaped, because the liver of a bird, right? Or we could still leave the shape aside. We could still say that that it is large, or it has a large surface area, guys, and it is used for what? It produces bile, right? It produces bile, which is stored where? Which is stored in the gallbladder, right? And we know that bile has special important purposes in what, guys? In digestion. Okay, so each time you're being asked to annotate the diagram, always make sure that you give a brief description of the structure before giving its function. Okay, because dissecting, annotating just one part, guys, can earn you about 3 marks to annotate just one part. Okay, just the fact that you can identify the part, that is 1 mark. Then giving a brief description, guys, is 1 mark. Then the function is 1 mark, a total of 3 marks. Okay, so guys, don't joke with your annotations. Okay, don't joke with them, guys. It's super important. And friends, don't forget this, guys. It's super important. Okay, it's super important. Take caution, guys. Don't ever forget this point, that the more parts you have on your diagram, guys, the more marks you get. Okay, the more parts you have, the more marks you get. Example, guys, if you're being asked to dissect a fish and display the respiratory and circulatory system, guys, you should know that for the circulatory system, you can be talking about the heart, right? The heart. But friends, in accordance to this point I said, the more parts you have on your diagram, the more marks you get. One mistake that students always make is that they just come and label the heart and everything ends there. Friends, that's wrong. Okay, that's wrong. Don't do that. Okay, don't do that. Now, like I said, the more parts you have, the more marks you get. What I'm trying to say is that to label the heart of the fish, you must have talked about what, guys? You must have talked about the sinus venosus and what the conus arteriosus, the ventricles, the auricles, and so on and so forth, guys. You don't just label the heart and say the heart will end there. They are going to mark it wrong. Okay, they're not going to give you the mark. So you should rather go to the fish. You can talk about the blood vessel that brings blood into the auricles. keep in mind, friends, keep in mind that the fish is a two-chambered animal, right? Because the heart of a fish has just two chamber. It has just one auricle and one ventricle, and the blood vessel that brings in blood into the ventricle or into the auricle, guys, is called what? It's called the sinus venosus. Okay, you have to name it separately. That is sinus venosus. Then to annotate it, guys, you can simply say that what it is cylindrical, or it's what, tubular, right? You simply say tubular, comma, brings in blood into what? Into the auricle, right? Good. It brings in blood into the auricle. Great. Then conus arteriosus, you can see that it is still tubular. Good. It carries blood where, guys? It carries blood from the ventricle and distribute what to all parts of the fish, right? So friends, please, if you're not subscribed to our channel at this point in time, guys, don't forget to hit that subscribe button so that you have access to all. There are so many contents in our channel available, guys, for you. So many contents, and the way in which you can remedy most of these issues, guys, just go to our channel. You are going to find so many interesting, great contents for you that can help you as well, guys, so well in all the subjects in both advanced level and ordinary level. So guys, visit our channel, invite your friends so they can pass as well as you. If you pass alone, guys, and your friends fail, that will not be good. Okay, good. So friends, I hope you've taken note of all these points I've listed. You should always, like for instance, guys, you can be asked to annotate or to label the structures of the fish that have to do with respiration, right? You know, the gills are the one playing those roles, and you should not go to the gills and label it as a gills. No, friends, label it part by part. Okay, the gills has a gill recus, it has a gill filament, it has a gill arc, and so on and so forth, right? So label each of the parts as the gill arc, the gill filaments, and so on and so forth. Good. So friends, that's how you can do to avert that particular issue, guys. So friends, I hope all of these points were quite understood, right? Good. So let's now move on to the last question on that paper, guys, which is a question which is based on experiment. Okay, so friends, for experiments that involve osmosis, you should always use the keywords like hypertonic, hypotonic, and isotonic, and so on and so forth, guys, because these are important biological words that are required of you to use. Okay, like for instance, let's say for instance that you put a potato tuber in a solution, right? After some times now, and the length of the tuber reduced, or the tuber became flaccid, right? What you say is that the potato tuber solution is hypotonic to the solution, because water left the tuber into the solution, right? So now the tuber in this case now is regarded in this case as what? As the weak solution. Okay, so make sure you use this word because they are going to give you more marks. Someone that uses these words appropriately and correctly is going to score more marks than someone who did not use it but have all the points. Okay. And please, friends, just for an advice, if you're not so versed using the words, right, it is better not to use them than to use them wrongly and instead put more problems on yourself. Okay, so you should practice how to use them so well. Good. Then friends, for control experiments, you should always use heat, as it gives a counter expectation, guys, because you can be given the reductase enzyme, right? They can give you fermented palm wine and ask to carry out an experiment to show the effect of the reductase enzyme on methylene blue, right? After doing it so well, they can ask now to describe a control experiment for it. That's why I'm saying that you should always use heat, because with heat is going to kill the enzymes present in the fermented palm wine. It kills the enzyme, and hence no methylene blue is going to be decolorized. Okay, so heat is the easiest experiment that you can always use as the control experiment. Good. Then guys, for the food test, you should always learn the various reagents used for the presence of the various classes of food and their expected results. For instance, guys, if I'm asked to test for starch, my expectation or the reagent in which I'm going to use, I know that I need to use iodine solution, right? I must use iodine solution. If I transfer, let's say, 2 ml of the test solution in a clean test tube, I'm going to add 2 ml of iodine solution in the test tube. And guys, my expectation, I expect to see a blue-black color before I can conclude that starch is present, right? If I don't see a blue-black color, I don't conclude anything. Good. It works for all of them, guys, as well. And so, guys, for the purpose of time management, guys, not all experiments need that you must carry them out. Not all. For instance, they can ask you to, they can give you, or they can ask you to remove some organs of the organisms you dissected. Let's say you dissected the mammal, right? And they can ask you to remove the liver and macerate or you pound. But guys, use the word macerate, and ask to macerate the liver, macerate the muzzle, and macerate the fur, right? Let's say we are macerating the liver, the muzzle, and the fur. They ask you now to add hydrogen peroxide to determine the amount of catalyst present. That experiment for me, guys, I won't carry it out because I know that what is going to give me the highest amount of foaming is the liver, right? Seconded by what, guys? The muscles, and third by what? The fur. So I won't carry it out, guys. I'll rather report the experiment. I'll report it because I know what I must see and what I'm going to see normally. Good. And friends, this is super important, guys. This point is super important. Always report your experiments, guys, in past tense. Okay, don't be, because there are some students that will enter into the lab and they'll ask them to describe the experiment. They'll say, measure that, do that. No, guys, always use past tense, like 2 ml of the solution was transferred into a clean test tube. Okay, I use the word was, right? And was is what, guys, is a past tense, right? Good. So always report all your experiments in past tense because you also tell the examiner what you did and you did in the past, right? Good. So always report them in past tense, guys. So guys, I think we've gone through all the hints that are so guaranteed for you to score your A grades, right? Good. And friends, please don't forget to subscribe to our channel if you want more interesting contents. And good luck to all those who are taking biology this year. And guys, watch out for the secret to grab the paper 3 physics with your left hand without stress, guys. So friends, after this video we shall upload that for physics, how to grab paper 3. So guys, subscribe to the channel so that you have access to watch it, and stay safe, guys, and good luck as you prepare for your exams. The remedy to all of these points I've listed, guys, is found in our channel. We have so many videos. Explore them, guys, and pass your exams. Thank you for your patience, and have a blessed and great day.